Hello and welcome to Microbiology Shorts. These are short videos on microbiology topics. My name is Rebecca Payne, and in this topic we're going to talk about the life cycles of bacteriophage. Bacteriophage are, of course, viruses that infect bacteria. There are two different life cycles that we'll talk about in this short. One is a virulent phage life cycle, and one is a temperate phage life cycle, but really we subdivide the life cycles into what we call the lytic cycle and the lysogenic cycle. So virulent phages can undergo the lytic cycle, where temperate phages can undergo either the lytic cycle or the lysogenic cycle. What happens during the lytic cycle, which both type of phage can undergo, is that the phage is going to infect, it's going to replicate inside of the infected bacteria, and then it's going to lyse that infected bacterial host cell. The host cell will then burst and die, and so the lytic cycle is a very destructive cycle for bacteriophage. Temperate phages either undergo the lytic cycle or what we call the lysogenic cycle. The lysogenic cycle happens when phage DNA is inserted into the host chromosome and it sort of hangs out for a while. There are no new virions made, new small virus particles, and there's no lysing of the host cell during the lysogenic cycle. The lysogenic cycle can happen for the life of the bacteria, and that phage can get replicated as the bacteria replicates during binary fission, or there might be some unknown cue that causes that inserted phage to excise itself from the host chromosome, and then it goes back and resumes a lytic cycle again. So those are the two cycles that we'll talk about here. We have an animation for viral replication, virulent bacteriophage, and then viral replication, temperate bacteriophage. I'll play those back to back here in just a second.
Virulent phages carry out a simple life cycle called the lytic cycle. The phage attaches to the host cell during the attachment stage, and then penetrates the host cell and injects its DNA during the penetration stage. After penetration, the phage causes the host DNA to break into small pieces. The phage then uses the host machinery to synthesize new copies of its DNA. This process is part of the biosynthesis stage. Biosynthesis also involves production of viral proteins. Once biosynthesis is complete, the phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages go on to infect other cells. Temperate bacteriophages carry out two types of life cycle, the lytic cycle and the lysogenic cycle. The lytic cycle for temperate bacteriophages is similar to the lytic cycle for virulent phages. The phage attaches to the host cell during the attachment stage, and then penetrates the host cell and injects its DNA during the penetration stage. In temperate bacteriophages, the phage DNA forms a circle, which can either replicate and be transcribed to produce phage components in the lytic cycle, or can proceed to the lysogenic cycle. During the biosynthesis stage of the lytic cycle, the phage DNA directs the host cell to synthesize viral components. The phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages can go on to infect other cells. Recall that temperate phages can proceed either to the lytic cycle or the lysogenic cycle shortly after penetration. In the lysogenic cycle, the phage DNA integrates within the bacterial chromosome by recombination. The inserted phage DNA is called a prophage. Most of the phage proteins are repressed by two repressor proteins that are products of the phage genes. Whenever the bacterium reproduces, the prophage is also copied. The prophage is excised from the host chromosome in a process called induction. Induction can occur spontaneously through recombination or some other genetic event, or through the action of UV light or certain chemicals. At this point, the phage may enter the lytic cycle.
So the lytic cycle of a bacteriophage involves 5 separate steps. The first step is attachment. The phage, which in this case we're looking at a bacteriophage that kind of looks like the lunar landing capsule I was describing in lecture. It has a head, it has tail fibers, and it has a needle-like injector that it can poke through the cell wall and insert the DNA inside the cell. So during attachment, the tail fibers attach to the outside of the host cell, exterior to the cell wall. During the penetration stage, the phage, which carries its own enzyme called lysozyme, and lysozyme is designed to attack the peptidoglycan in a gram-positive, gram-negative or acid-fast cell wall. The lysozyme is able to break apart the cell wall a bit. The tail sheath, which is the injector part, contracts and it forces DNA into the cell. In the third step, biosynthesis, you get production of new phage DNA and new phage proteins. And of course, the phage is using the bacteria like a factory for itself, because the phage itself does not have any machinery to put together new proteins, doesn't have any ribosomes of its own, it doesn't have any DNA polymerase of its own. So it's using the host cell's enzymes to put together new DNA and new proteins. Maturation happens then, which is assembly of the new phage particles. This generally happens in more or less a spontaneous manner. And then release happens when the phage lysozyme again breaks the cell wall and millions of new virions are released. Usually in the release phage, the cell wall is broken and the bacteria itself is lysed and killed. The definition of a virion is an infectious virus particle. So each virion that's made inside the cell then goes and infects a new cell, making the cycle continue. The lysogenic cycle only happens in temperate bacteriophage, and temperate bacteriophage can also do the lytic cycle. So this is kind of a special addition to what temperate bacteriophage can do. During the lysogenic cycle, you could have attachment and penetration, the same as in the lytic cycle. But then at the third stage, instead of having new DNA create new proteins, instead you have integration of the DNA into the bacterial chromosomal DNA. When the bacteria reproduces, as it does by binary fission, the phage DNA is also going to get reproduced and transmitted to the daughter cells as a piece of the bacterial chromosome. This can happen for generation after generation, thousands of generations, or it can be permanent. But in some bacteria, at some point there may be a cue that causes the DNA to excise from the chromosome. It becomes again a circular piece of phage DNA, and this begins the lytic cycle once again, and it goes back to lysing those bacterial cells and creating new bacteriophage. There are some special properties that happen with lysogeny, or the lysogenic phase. One is pathogenesis. If you call the inserted bacteria prophage, this prophage often gives the cell immunity to reinfection by the same phage. Not immunity to infection by a different phage, but say it was infected by a T-even phage like T12, then you're not going to get another T12 bacteriophage able to infect that same bacteria that has a T12 prophage. There's something called phage conversion, which causes special properties to be conferred on the host cell. During conversion, which is a type of a state that the bacteria is in when it has a prophage, you can have pathogenesis conferred by the prophage. I've got 3 examples. One is Corynebacterium diphtheriae, one is Streptococcus pyogenes, and then there are other Streptococci species that have this phage conversion property that gives them virulence. In Corynebacterium diphtheriae, we know that Corynebacterium diphtheriae can cause the disease diphtheria. It's part of the childhood vaccination series. Diphtheria causes a pretty acute, sometimes fatal, childhood illness. But Corynebacterium diphtheriae can only be pathogenic when it's infected with a lysogenic phage. And this phage has to contain the gene for the toxin that confers virulence. And virulence is a vocabulary word that basically means the ability to be pathogenic. Streptococcus pyogenes can only cause scarlet fever, which is one of the many diseases that streptococci can cause. Pyogenes, the specific species, can cause scarlet fever, and among other things. But it only causes scarlet fever when it's lysogenized by a bacteriophage T12. So it has the T12 inside its DNA as a prophage. This T12 carries the SPA gene, and SPA is able to produce another toxin that causes scarlet fever. Other streptococci species, if they carry, again, the same T12 bacteriophage, can cause a disease called toxic shock syndrome, which is not uncommon and wasn't uncommon in the early days of tampon use. But streptococci can only cause toxic shock when carrying the T12 phage, again with the SPA gene. So lysogeny is interesting. And disease by bacteria often requires not just the bacteria themselves, but also an integrated bacteriophage genome that confers special toxic abilities to the cell and allows them to be a pathogen. That's the end of this microbiology short. I hope you have enjoyed this subject of bacteriophage and the lytic and lysogenic life cycle of bacteriophage. I hope you join me the next time. And for now, have a great day. See you later.